So welcome everybody to the Sachs Biotech in Europe Forum and the Advanced Therapies in Immuno-Oncology panel. My name's Sarah Holland. I'm the Chief Business Officer of VectivBio, and it's a great honor to chair this panel today with a group of illustrious panelists. I'm delighted it includes 3 female CEOs. I think that's a great step forward. And I'll hand over to my co-chair, Esteban.
Thank you, Sarah. I am Esteban Pombo-Villar. I'm Chief Technology Officer of the TargImmune Therapeutics. And it is really a pleasure today to host and co-chair this very illustrious panel. I would ask the panelists to provide a brief introduction to themselves and the company. And why don't we start with Kristen, please?
Great. Thank you so much, Sarah and Esteban. So it's great to be here today, and I look forward to hopefully moving back to in-person meetings and conferences. But so I'm Kristen Albright, and I've been recently appointed the CEO of Precarium. I bring about 15 years experience to the organization, initially starting my career in large pharma and having spent both time in mid-sized biotechs and venture capital. So at Precarium, our goal is to be a leader in the field of microbial immunotherapy. And more specifically, we're leveraging the biology of a non-commensal human-specific pathogen, so a bacteria, that has natural tumor homing and oncolytic abilities as a therapy for solid tumors. Our lead program focuses on locally administering the bacteria intravesically as a monotherapy for the treatment of non-muscle invasive bladder cancer, while our pipeline goes a step further via integrating immunomodulatory cargo into the bacteria's genome to improve immune activation in the tumor microenvironment. To put our mechanisms simply, even though it's quite complex and still not known, what we've seen so far is our bacteria can recruit cross-presenting dendritic cells and facilitate antigen spread and T-cell recruitment. So really altogether, our goal is activation of the desirable immune responses.
Great, Kristen, thank you. Thank you very much. Natalie.
Thank you very much. So I'm Natalie Mount, and I'm the CEO of Adaptate Biotherapeutics. So at Adaptate, we are a next generation immune-oncology company, and we develop a portfolio of therapeutic antibodies. And our antibodies target a particular immune cell subtype called gamma delta T cells. And particularly, we are interested in the delta 1 subtype of gamma delta T cells, which is a really intriguing cell type because it's a tissue and tumor resident cell type. So we have a portfolio of antibodies which are now progressing. We actually started Adaptate nearly 2 years ago now, when we spun out from Gamma Delta Therapeutics, where we started our work. And we've had backing during that time from both Abingworth and Takeda Pharmaceuticals. So we are still a preclinical stage company at this point, but moving very fast now towards clinical translation, particularly for our lead approaches, which we hope to take into the clinic for solid tumors.
Great, thank you. How about Susan?
Good morning. It's a pleasure to be here. Thank you very much, Esteban and Sarah, for inviting me. So my name is Susan Hill. I'm the CEO of Mestag Therapeutics, and Masstag is a company which is looking at the role of fibroblasts in modulating the immune system in disease. So many people will be aware of fibroblasts from fibrosis and wound healing. We're actually looking at them through a new lens. We're looking at the role of fibroblast subpopulations in modulating immune effector cells in the disease environment. We're able to do this through single cell level analysis that has been conducted by our founder team. And they've helped to create a new framework for the role of these cells within the disease environment, whether it's in inflammatory disease or oncology. So as a company, which we founded just over 18 months ago, we focus across both therapeutic areas because we believe there's commonality of biology here between immunology and immuno-oncology. We recently extended our seed round to 45 million, bringing on board investors including Google Ventures, North Pond, Forbion, in addition to JJDC and SV. And we're working on progressing a portfolio of first-in-class antibody therapeutics. And we're also working closely with J&J's immunology division, discovery organization, on a target discovery collaboration.
Thank you very much, Susan. Søren.
Yeah, thank you, Esteban. Thank you, Sarah. It's a pleasure being here, contributing to to the SACS Forum. I'm Søren Breinhard. I'm the CEO of Alligator Bioscience, a Swedish publicly listed biotech company. We are listed at the main market at Masstag in Stockholm. And Alligator Bioscience is an antibody company developing best-in-class both monoclonal and bispecific antibodies in immuno-oncology. Our lead program is just on the brink of recruiting the first patient in a phase 2 study in pancreatic cancer. And we're very excited about that. And we have a deep and broad pipeline with proprietary programs in preclinical and various clinical stages, and also a number of partner programs. And our new and very exciting technology, Neo-X Prime, sort of brings the last 20 years of Alligator sort of together in one program by combining our huge experience in CD40 agonistic antibodies with our antibody technologies, making a oncology principle here where we agonize and stimulate dendritic cells, while at the same time recruiting exosomes from the tumors to the dendritic cells for antigen presenting and T cell activation, thereby really sort of giving a boost to the patient's own immune system.
Great, thank you. Eric, please.
Yes, thank you, Esteban. So, hello everybody. My name is Eva Liwa. I'm the president and CEO of PDC Line Pharma. I've been involved in several biotech over the last 20-25 years. Related to PDC Line Pharma, we are a cancer vaccine company, clinical base. We have currently a phase 1-2 ongoing in lung cancer, non-small cell lung cancer in different countries in Europe. We raised close to 35 million euros of financing, and we're in fact in the process to close an expansion round of the B round of financing that we hope to announce in October. The technology platform we are developing in the company is based on a unique plasmacytoid dendritic cell line coming from the blood of leukemia patients having a very rare leukemia PDC, which is the specific part of the dendritic cells in the blood. And we use this cell as a vaccinal platform by combining with GMP peptide representative of antigens to develop cancer vaccine. So that's the company. We're 35 people based in Belgium and France.
Thank you very much, Eric. And Daniel, please.
Yes, good morning. It's very early morning for me. I'm Daniel Tepper. I'm the principal founder, chairman and CEO of Cytovia Therapeutics, which we started a little bit over 2 years ago. Actually started my career in Basel, Switzerland at Novartis, but most of my career has been in the US and currently based in Aventura, Florida, which is north of Miami, where the headquarters of Cytovia are. So Cytovia focus on natural killer cells. And we have 3 core technologies. We have the ability to differentiate NK cells from induced pluripotent stem cells, iPSCs. We have gene editing capabilities through our close partnership with Cellectis. And we have immune cell engager antibodies, which we can use on their own, but more interestingly in combination with the cells. We have diversified pipeline with a primary focus on solid tumors, and our lead program targets GPC3, with the lead indication being hepatocellular carcinoma. We also have programs in glioblastoma and multiple myeloma. The company started as a virtual play with a number of academic partnership, not just in the US, but also in Europe and in Israel. And we've recently, say in the last 6 months, become much more brick and mortar. As I said, the headquarters are in South Florida, but we have a very strong research team close to Cambridge, Massachusetts. We have GMP cell manufacturing facility in Puerto Rico that will fully open in early next year, and manufacture the batches for clinical trials, which are expected to start in the spring, summer of 2022. And we just announced last week the formation of a joint ventures that focuses on development and commercialization in China. So this is a new company called Cytovia, where we have approximately 40% of the ownership of this company. And in conjunction with that transaction, we raised $45 million. We anticipate to be public in the US in 2022.
Great, Daniel. Thank you very much for waking up so early to participate with this.
Always a pleasure.
Today, it's great. So let's bring it back to Basel and make the connection with Daniel back to our headquarters here in Basel, where we have been thinking for this panel on how important these advanced therapies are, really the immune system and controlling, modifying, and harnessing the power of the immune system to provide clear benefit to patients that definitely are in high medical need. So from hepatocellular carcinoma, pancreatic cancer, we have heard really the promise of this technology. One very important aspect, of course, is how do you stand out from the crowd? I mean, sometimes you hear from investors and pharma, perhaps a little bit of tiredness of hearing about the promise of immuno-oncology. And I'd like to think, with the team, what do you think about that? Isn't this promise getting a little tired? How about Natalie? What do you think?
I think it's it's a really important question, Esteban. I guess the way I think about it is immuno-oncology, that promise. I mean, actually, it's already delivered a lot. We've made great progress in this field. And you think actually, immuno-oncology has brought new treatments for a whole range of cancer indications and really revolutionized actually therapy in a whole range of indications. But I would characterize it as, yes, we have delivered on that promise, but actually, the job is not done yet. There's still more work to be done. So if you look, for example, there were patients we all know who don't respond to these current therapies, but also if you think about checkpoint inhibitors, we know that patients who do respond initially, eventually they will go on to relapse. So there is still a really compelling unmet need there. So I think for investors, the good news is immuno-oncology has delivered, but for those of us now working in the next generation, I guess our promise is, yes, there's more that can be done. There is an unmet need that needs to be solved for patients, and we can bring new therapies forward to that. And I think what we can do really is to make sure we build on what we've learned. I think from the existing therapies, actually, we've learned a lot about the cancer immune system, about the immune responses, and about different tumor types. And now we need to apply that in a new way. And I guess that's why companies like mine, Adaptate, we call ourselves a next generation immune therapy company, because what we're doing is building on what's been done in the past and taking that forward in a new direction. So, for example, in our case, we think about people have been looking at how to activate T cells. So we've got CD3-based approaches in the clinic, which are pan T cell activating approaches. What we've been doing is thinking about, well, how do we learn from those? What's worked in that area? And actually, how do you apply that then in a new direction? So in our case, what we want to do is, rather than having a pan T cell approach, we've decided to work on a very particular subset of immune cells, these gamma delta T cells. And in fact, more specifically than that, actually work on these tissue and tumor resident delta 1 subtype gamma delta T cells. So I think that's an example of how we can really apply what we've learned in the field from existing therapies and then take it in a new direction. Because what we've learned is if you move ahead with pan T cell approaches, actually there are a lot of side effects that go alongside that. If you can go with a more specific subset, then the idea is that you can avoid those side effects. But we know also it's really important to be able to work not just on a subset, but also to communicate with the general immune system, really, to be able to bridge, I guess, between the innate and the adaptive parts of the immune system. But also, particularly, we're really interested in solid tumors, and I think number on this panel also interested in solid tumors. And it's a fascinating scientific area, but an area where there's really great unmet need, and one where we've learned actually that you need to take new approaches. So, for example, people have been trying to use blood-resident cells in order to be able to treat solid tumors. So the approach we've taken is, well, actually, if you really want to get into a tumor environment, then how about actually using a cell which is already tissue resident, tumor resident, something that can really actually move into that immune tumor environment and be naturally primed to survive in that. So I think, to me, that's kind of examples of how we can bring this, to say, to next generation. I think that when you apply that next generation science, build on what's already been learned, take it in a new direction, then actually you can address unmet needs, which is really critical for patients. And that also provides, I think, a really compelling proposition to investors.
It's exciting that several of our panelists have actually raised money recently. So it looks as though investors are not yet exhausted by this space and see the opportunity and the unmet need. So that's great. I mean, I've been looking at immuno-oncology since working on IL-2 in the 90s and, followed cancer vaccines. So, we've been looking at immuno-oncology for a very long time, but do we even now understand the biology well enough to select the right patients, to choose the right mode of action, to combine things in the right way? I mean, maybe I could ask Susan to comment on where we stand on the biological understanding of this area.
I love that question, and it follows on so nicely from the last one as well, because I think the key to standing out from the crowd is having an approach that hasn't been explored before. That's a new way of looking at this, and one of the things that has made great strides over the last few years has been the ability to look at things at a single cell level, which is exactly what Natalie and Adaptate are doing, and it's also what we're doing at Mestag, which is to try and understand within the complexity of the tumor microenvironment, where there are multiple different cell types. It's not only the kind of the high level types of cells, T cells, B cells, tumor cells, stromal cells. Within each one of those, then there are subpopulations, and each of the subpopulations are doing different things. And so in the old days, as it was, one would say a fibroblast was a fibroblast and a T cell was a T cell. And what we've learned over the last 20 years is that actually, there's a lot more subtlety than that, and dissecting out those roles can be extremely powerful from a therapeutic perspective. So do we feel like we understand it fully now? We know everything? I don't think so. It's massively complicated, as everybody around the table knows. And I think, the fact that there's such a diversity of different approaches that are in exploration, where we're trying to learn from previous studies and incorporate not only the learnings from the trials, but also the new technologies, ways that we can identify mechanisms, pull them out from a general patient population, or look at them carefully in animal models to be able to understand them. These are real advances, and they're quite, one I think has to be quite creative in thinking about how to leverage them going forward. So my sense is, we're still learning about all the different cell populations. We're learning about how they interact with each other and interact also directly with the tumor cells, how they influence availability of antigens, so that you not only have all the cells in there, but you also have the kind of kick, so that the right cells are recognizing the right antigens and then activating. So there's a lot going on. And I think actually, we're starting to make great strides in untangling this and understanding it further. But ultimately, where does it go from a development perspective? It probably goes further and further into more and more patient segmentation as we understand this further, and then the tools and technologies that go with that. So can we actually translate that from a sort of mechanistic understanding then to a clinically identifiable patient group? I think there's some really exciting progress still to be made and some challenges to be overcome there, that's for sure.
That is clearly really the next step, because having this fantastic new science and insight into biology and developing some really cool technologies to modify that and get perhaps some nice in vitro or preclinical data that suggests this is a very fascinating novel mechanism. But really the question that you highlight there is really, how confident are you, or are we, that the results will translate into patient benefit, and why is that the rationale? How would you demonstrate efficacy or comparison to standard of care, or which animal models, for example, are credible or useful to help predict this benefit? And I know we have all struggled and thought about this process, but what about Kristen? What do you think about this?
Yeah, I mean, you gave me the $1 million question, right? I mean, yeah, as Susan mentioned, biology is so complex, and not just within each indication, but each sub-indication, and you could continue to go down that rabbit hole. But, and even just given the statistics alone, the probability of all of us on the panel is against us, of us actually translating what we're doing into the clinic. But, I'm a firm believer that if you limit yourself to that sweet 18-month window or so to challenge your hypotheses in key preclinical experiments, that if you can make it through that and you're asking the right questions for every experiment you're running, and you come out the other end, then you have a solid package. And the package is mostly described as compelling or convincing, although there's actually no way to objectively measure what compelling and convincing are. I would say one recent example at Precarium was we had a hypothesis. It was kind of a crazy hypothesis that if we test it, and given our bacteria systemically, so single oral dose systemically, prior to administering our bacteria locally or intravesically, would that actually increase the anti-tumor activity and survival in preclinical models? And we ran this experiment in a very well-known orthotopic MB49 model for non-muscle invasive bladder cancer. And what was so interesting and repeated was that we saw a large, clear benefit of adding, again, this systemic type of immune response prior to administering a therapy intravesically. And as you mentioned, Esteban, it was also against the standard of care that's currently BCG for muscle invasive disease. So what's interesting about this experiment was it completely changed what we decided to now look at for our IND filing next year and how we want our phase 1, phase 2A trial to look. And it's, I think, a great example of leveraging some key preclinical data on not only decision-making, but designing of your phase 1 study and bringing a product or program into the clinic. But we all have to realize that, translation doesn't stop there. It's also getting your biomarker package together, your translational package while you're getting your tissue samples in the phase 1 trial and throughout early clinical development to make sure that the patient population that's going to benefit the most from your therapy is the one that you're going after. But yeah, it's an interesting question. And I think we could sit here all day debating on which models are the best, but yeah.
Does anyone have a comment about using animal models with humanized immune systems versus other sources of data like ex vivo human cells or using translational medicine? Natalie, maybe you had something.
Yeah, I think it's a really challenging area, and as Kristen was just saying, I think exactly as you said, it's really about choosing the right model and the right experiments for the question that you want to answer. So you need to define what that question is, and I think we all recognise that you're not going to be able to make an animal model that actually truly represents what's going to happen in every cancer patient in your future trial. That's not going to be possible. So it's then about actually dissecting down to find the right model that actually enables you to answer a specific question. In our area, for example, the immunology is so different between, for example, a mouse and a human that actually doing mouse biology is that's going to be very limited, if any, utility. So actually, now with new tools, it's more and more possible really to focus those questions more on a human system. Mm-hmm. So, for example, access to human tissue and trying to make the most of reverse translation, moving back from actually what people have observed in the clinic over the years, what actually, and then moving into being able to get actual patient explants, for example, and be able to look at particular parts of your mechanism in there. And I think to me that's what you're trying to do, is actually make sure that you've really got a solid mechanistic hypothesis that you understand as much as you can about your therapy in a setting. But then after that, really it's a question of then moving towards the clinic, as Kristen said, that's really the way you answer these questions, is in the end to be able to move towards the clinic and try and select the best patient population you can to be able to test things in a clinical setting.
And it's interesting what you said. So back translation, isn't that how gamma delta T cells were actually discovered, was from clinical observation? Is that, am I right in understanding that?
So I think the recognition of actually the role of gamma delta T cells, and reverse translations played a really big role in that, because people have been looking at, for example, doing bone marrow transplants in patients, and people know you have to deplete alpha beta cells from those transplants to avoid graft versus host disease. But actually, you can maintain a gamma delta population when you do the transplant, and then they were starting to realize that actually, when they followed those patients, the patients who had more engrafted gamma delta T cells actually had a better survival than those who didn't, had a much smaller engraft population. And then you kind of build out from that. Other clinicians then started to look at patients and started to build the correlations of who does better over time, and actually determine that gamma delta T cells and these delta 1 cells are really important as a positive prognostic factor. So I agree, that kind of reverse translation is really, really critical, and we won't ever get that from a mouse model. That really is from human learning and understanding the human system.
We really need to build a compelling reason to believe based on multiple sources of argument and data and evidence. So you talked about clinical, maybe moving on to the crucial clinical stage. How do we do clinical trials in immuno-oncology when the standard of care is so complex and so expensive, and there is so much we're still learning? Maybe I could ask Susan to maybe share some insights from working in the rare disease space and how that might give us some learnings.
Yeah, no, that's a great question. And I think, this is where new technologies that are often being used at the moment preclinically, we need to see them move towards clinical application. And when especially when you're an early discovery stage company and you're sort of projecting ahead into the clinic, you want to be able to project ahead not only what the standard of care might look like in X years time, but also what the technology might be that could be available then, so that you can identify patients and you can monitor them during clinical trials. So interestingly, as Natalie was talking, I was just reflecting on some of the work that led to the foundation of Mestag, which is that it came about through single cell analysis of human tissue samples in pancreatic cancer and in inflammatory diseases. So we sort of started with the human, went through the mouse, and then came back to the human again. So being able to incorporate that kind of level of granularity into trial design. So, for example, thinking about patient selection, the sort of historical levers in the tumor space have been, tumor genetics, or they've been, target expression. They might now be things like cell type, sort of composition of the tumor, or kind of the way the tumor is organized, for example. And so then you get into things like, needing to take biopsies routinely, starting to look at, different subpopulations of cells in any one patient. So, these things are coming. They're not necessarily sort of fully rolled out and used as standard in clinical trials. But I think the closer that we can get to aligning the mechanism and the patient, and this is back to your comment on rare disease trials, of course, this is, where you have a single mutation or a single lacking protein or whatever in rare diseases, you get that beautiful, that really exquisite kind of lining up of mechanism and therapy. It's just much harder to get to that point in the tumor environment. But I think that's the direction that we're all going in, and we're all sort of trying to strive to. So, and connected to that is, how can you leverage new technologies to identify patients so that you can actually do your first trial in the patient population that you want to get to. So, oncology historically, because of the nature of the therapeutics that have been used to treat tumors, quite often the sort of paradigm is to start in late stage, very advanced patients and gather safety data. And you kind of have to go through multiple steps before you can get straight to your patient population. But of course, the ideal would be if you can identify your patient population, and you've got a kind of credible sort of proposition for your therapeutic to be able to go right there. So there's some sort of efficiencies, I think, of trial design, that there's kind of how can we kind of catch up on the lag of historically how trials have been designed versus in the future, how could we do them if we can start to incorporate these kind of technologies and start to sort of incorporate new therapeutics where you're getting down to a single cell level. So we're quite excited about that. We don't claim to have all the answers by any means, but we're excited about the challenge of, being able to do this.
And it's hard to look for a signal in a patient population that's been quite battered and whose immune system may not be fully functional by that point. And you're maybe trying to look for a signal in a population that doesn't have the ability to respond to your therapeutic. So I think your points are really important. Yeah.
And people always look for a signal, even if a study is just meant to be safety.
Exactly.
Certainly, and there's also if you want a philosophical, ethical question that comes across when you start discussing your first-in-man study, which is really you're going to expose a patient to a drug. Should you start with a dose that is absolutely super safe and will have absolutely no possibility of benefit to the patient, or should you already start with a way that provides some hope of potential benefit to the patient? How do you move from there? And if you do that, then the patients are probably those that could potentially benefit. So the philosophy behind that is a very important differentiator. So this
and
really,
I think
go ahead.
No, just one final again. It's the evolution of the field from a chemotherapy-based approach where you're killing all cells, but you're hoping that you'll kill the tumor cells more, if you like, to actually you can get the sort of safety, the risk benefit right, because you've got such a targeted mechanism. And so it'd be fabulous to be able to do that, to go straight into a patient population where you can both test the safety and you can see some initial signals in the right patient population straight away.
Certainly, back here I still have my selective toxicity from Albert as one of the textbooks of the last century. So really, that's very important. This technology advance and understanding in the field and personalized. So very critical is to then just perhaps change gears slightly. As we discuss, we've all raised funds, or we're in a process of raising funds recently, and we have lots of good news about that. Then for all the members of the panel, important is how do you fund the next stage of the company? Because clearly getting this great science is important, but what is essential in a business case for a VC or an investor at this stage? What is the data they must see to invest, right? You talk to some people, there's different opinions and different stages. For example, Søren, you've been in this field for a long time, and you've seen currently in Alligator and in previous companies many different perspectives in this area. What is your view on this?
Yeah, I think that's a very, very interesting question, Esteban, and unfortunately, it hinges up very much with the discussion that we just had, and the initial question that you asked about the investors being tired and impatient, and I think to some extent they are. We have seen the great progress of the checkpoint inhibitors, and I think a lot of the early investments were sort of based on that promise. I think if we look back in history, we can look at rheumatoid arthritis and Humira. I mean, big breakthrough. We could look, if you go 100 years back, on diabetes and insulin. And why do I mention those? Because that's sort of breakthroughs where you hit sort of a central player in that pathology, and that's exactly what happened with the checkpoint inhibitors. That we were hitting one sort of major switch that sort of brought us from no response to a lot, and now we are into sort of now trying to go deeper and deeper into individual patients and try to translate that. And I think for a clinical stage company like Alligator, I mean, when you're going to fund your company and, for instance, fund your trials going from phase 1 to phase 2, then what we look at today is data from, what can we say, normal basket trials in last resort patients, and we are trying to sort of build an investment case around signals from these patients. We might even take strategic development decisions on a partial responder in a certain tumor type in a large basket trial. And if we just take one step back, I mean, and we all have statistics. There's probably also a statistics book in your bookshelves back there. I mean, basically, we all know that it doesn't make sense. Mm-hmm. But that's just where we are. And I think what investors want, I think, is they want a translational story. And I'm sorry to say, doing in vivo studies in mice, even though we know that it translates so poorly, is most often sort of a part of our education, and what investors need to see. We need to provide a suitable hypothesis on how that sort of translates to the clinical data that we have, or that we have when we talk to the investors, and how that will eventually translate into clinical trials and patient benefit. And I think that 50% of that is data. And the other 50% is the people around the companies and the given assets. So I think it's a mix there, Esteban. We need to be credible. We need to take clever decisions, and we need to be able to tell our story. And I think that's what sets us apart. And of course, then sometimes you have molecules with fantastic data where they speak louder than most, but that is actually when we get into early clinic, that is few and far between, mm-hmm. in my view.
So the clinical data is fundamental, but the promise of the biology and understanding of the biology and the strength of the biological hypothesis data is absolutely critical. So maybe just to pivot to that, of course, one of the groups that we're interested in is in investors, so that they come along with us to our journey, and we advance our products together and benefit patients, and of course have a return for the investors. But the others are, of course, partners that can really commercialize this and bring our great inventions and benefits for patients to many different patients. Daniel, what is your view on partnering? What is pharma looking for these days in this space? What is the preclinical or clinical data that they are looking for, and what is the ideal stage to do that? What is your view?
Well, I mean, I think as was mentioned before, the pace of innovation is accelerating all the time. In our field of immuno-oncology and specifically cell therapy, let's just remember that the first autologous CAR T cells are what, 5 years old, and it's already looked by some as ancient history, because you go from autologous to allogeneic, you go from allogeneic from leveraging stem cells and gene editing and soon in vivo gene editing. So others pharma keep pace with the innovation, because you have to have different sets of scientists. Do you retrain your people? You hire new people? You acquire a company every year? That's becoming quite complex. I mean, the other dimension is, if you look at pharma in the last century, let's say in the 1990s, or just over 20 years ago, the big story was about managing risk and focusing on large markets and on incremental benefits, and it has worked to some extent in a number of therapeutic areas, but that's not the case anymore. I think today, and in part because of market access, everybody is focusing on disruptive innovation and on having a clear impact. Now, when you bring technology and clinical application together, many people have been trained that if you have a new technology, you should really test it on a known target, because you don't want to multiply the risk. The only problem with that is that you end up with 10 or 15 companies going after CD19, just to take an example of BCMA, and the question is, what's the ultimate target product profile, and aren't you going to disappoint investors, or are you going to get pharma to maybe license early and then realize that the target product profile is not competitive, so they will deprioritize, the product will go back to you, which investors and the market in particular don't like so much. There was also a mention earlier on, do you focus on basket trials of last resort patients, and which translates into very small market opportunity and generally speaking very marginal effect. I mean, we see it ourselves in hepatocellular carcinoma. Initially people could say go to the last resort patients, except that the ability of any new technology or therapeutic approach to prolong life at that stage is minimal. On the other hand, and if you look where the real patient need, only 20 or 30% of patients respond to current therapy in hepatocellular carcinoma, and even the advances with checkpoint inhibitors, they're incremental and welcome, but they're not sufficient. So we see clinicians screaming for doing clinical trials early on. I mean, we're not talking about necessarily first line, but much more second and third line, which gives you the opportunity of having a meaningful clinical effect, clinical response, and meaningful prolongation of life. So if you bring all of those elements together, it's a question of, as a biotech company, where do you put your resources, and as a pharma company, when do you partner? So we believe that given the availability of capital, biotech companies should pursue not only preclinical but clinical development as far as they can, at the very least until clinical proof of concept, and maybe beyond, maybe all the way to the market. Be extremely selective on the indication, and making sure that their products have the ability to be first in class or best in class, and address significant and unmet medical needs. So where does that leave the room for partnership prior to a potential acquisition? I would say increasingly, because the biotechs have technologies that pharma, for the most part, doesn't master yet, is really joint development from a discovery standpoint. So maybe in a biotech company like ourselves, as disruptive cell therapy and gene editing technology that not yet most of the pharma have, but pharma has been working on particular disease, on particular target, and we can join forces. So what it does in the end is that it allows the biotech company to graduate to more mature development, and it gives the ability to the pharma company to access innovation early and to have genuine discovery to clinical programs that can make an impact.
Great, Daniel, thank you. It's certainly very important to consider this, because, as you said, the CAR-T space is already a few years old. It was claimed to be a big revolution, and there were these mega transactions that took place, and yet, well, some of the recent news are not as exciting. There are certainly challenges, particularly application to solid tumors is disappointing, not to say very challenging, and even some that were expected nice, large phase 3 clinical trials are not as exciting, or not as the data is not what is expected. So really, what is the next thing that we can expect? Is this really a space? Eric, what do you think?
I believe what's happening is really completely normal. You have a new technology, because I would say CAR T-cell is still considered as a new technology. It has been put on the market 5 years ago, and now, as has been said by Daniel, there is new incremental improvement of this technology with allogenic, with fully in vivo system. So to have what is very important to mention is that this CAR T-cell with the checkpoint inhibitor has been considered as a success, because they have been put on the market. They make significant sales, and they are considered as a mega blockbuster. So that was extremely important to redevelop the immuno-oncology. We just have to remind that 10 years ago, just to say immuno-oncology was not at all in the key trend. So now with all these new innovations related to checkpoint, to CAR T-cell, and the final validation, which is to be put on the market and to have a good pricing and good sales, has validated this overall concept. Of course, it's not. You don't have a magic bullet in science. So CAR T-cells has been working on liquid tumor. They don't have the microenvironment. They don't have the same issue that you can have in solid tumor. The complexity and the barrier to be effective is a lot higher in solid tumor. We know a lot of things now about what's happening, and I'm sure there's still a lot of other new science that will come to better understand this microenvironment and its evolutions with the stage of the disease. And I think it's where some new technology that are in fact in development today. I can talk about the NK cells. You have the gamma GT. We discussed the bispecific, the cancer vaccine were involved can be as well an interesting approach to be used alone or in associations. I believe today the association to treat cancer is definitely the way to go, and to have combo, to have triplet, to have 4 products, and according to the good segmentation of the patient will be very important. So even if we had some failure, even in phase 3 that happened, I don't think it changed today the mood of the investment, because there is still a lot of innovations that is extremely exciting coming, even in new checkpoint inhibitor, to target checkpoint inhibitor of the NK, for example, or of the gamma GT, or, to stimulate as well at the top of the T cells over part of the immune system. It's something very exciting. To boost the immune system independently of the checkpoint is as well something very exciting. So I believe there's still new waves that are coming and that will provide very good innovation in the future. So I stay very optimistic, even if we know we are in a risky business. Biotech, you have to manage risk. And we know that some of the projects, for different reasons, could be a failure. But money is there, good science is there, and maturity as well is on the good track. And manufacturing system has been improved a lot. Cost of goods of cell and gene therapy is really decreasing now with all the new technology and the new investment and the big players being present in this, the big providers in this area. So I think we are in a good place to be for the next few years and next decades. That's what I could say.
Maybe a brief follow-up question on that, Eric, because you said that the manufacturing is in place and the big players in manufacturing. Of course, antibodies are pretty standard, and you can get antibody manufacturing capacity huge. But in the cell therapy space, it might be a bit different. Does any of the panelists have an opinion on whether you should buy or build or partner in terms of manufacturing capacity? Daniel, do you have what do you think?
Sure. I mean, clearly, we're a young company, and we've made the choice to build. And actually, in the beginning, we were very cautious, and investors, rightfully so, said, if you don't build, you don't have a chance. And the pharma company that did not master the technology for the most part said that you have good ideas, you have good science, you have good experiments. If you cannot demonstrate to me that you can make it and you can make it GMP and you can make it cost effectively, we're not so interested in talking to you, or come back later. So, we've made the investment. I have to say that, one of the advantages of cell and gene therapy is that from a manufacturing standpoint, it's much more miniaturized, and it's actually, it's less impressive. So if you say, this is my manufacturing room, people are thinking about the huge bioreactors and, Amgen investing hundreds of $millions in Ireland for gigantic, plants. And is there, everything now is miniature. The machines are expensive, but they're expensive $500,000 or $1 million, each machine. So it looks more like a lab. It doesn't mean that it's not very sophisticated. And I think what we find out is that the CDMOs, even the most sophisticated CDMOs, are struggling to keep up with the innovation, and are basically saying, teach us how to do it, and then we'll do it for you. Except that transferring the technology and teaching them how to do it, cost you more than building your own facility, and obviously you're losing a lot of the know-how, because people file patents, but at the end of the day, from a manufacturing standpoint, most of the differentiation is in the know-how, and the real IP is even in cell and gene therapy is in the composition of matter, or in the specific effects, biologic effects. So I would say yes, in the business plan for a company that operates in cell and gene therapy, having its own manufacturing is critical. Something that we really stand for is not only advancing toward a cure for cancer, which we believe we can do with cell and gene therapy, but also improving the access. And improving the access for us as two components is having safe enough products that they don't need to be administered in the hospital or in the ICU, even in the case of CAR T cells, but also is lowering dramatically the cost of goods, so that a cell therapy product can be priced as another biologics, not as a $1 million adventure. If you put together the price of the drug and the associated hospital costs. So yes, manufacturing is essential to compete effectively in cell and gene therapy.
Thanks, Daniel. So it is clear, we have touched a number of very important topics. We started talking about the importance of understanding new biology and how the new biology is driving the innovation in a field that has just started. So yes, immuno-oncology has been used as a word that is common, and people have heard it many times, and there is really remarkable results in recent years in the ability to harness and direct the immune system to attack cancer and providing great patient benefit. But there's still a lot of room for innovation. There's still a lot of things that we don't know. And as we progress and we understand more, we need to see what are the relevant biological readouts that will generate patient benefit. And we will continue to develop our story and understanding to bring investors along with us to take that drugs to patients, and to bring forward all the excitement in this field that has only just started, from the cell therapies to the antibodies and small molecules that can modulate and transform the tumor microenvironment, even directed cell killing combined with aspects of real stimulation, selective populations of the immune cells. That is all really new and promising. And again, we're very excited about this field. And what we have also heard is that the new technologies really pose also a manufacturing and an access challenge. And we should not forget that if we make a great therapeutic breakthrough that is extremely difficult to manufacture, it will not reach all the patients that could benefit. And if we make it such that it's so complex and so expensive, it will not reach all the patients that it will benefit. So really, in this short period of time, we have covered many aspects about the new advanced therapies and how really, I would say, this is just the dawn of immuno-oncology. We're just starting to scratch the surface. And it's very exciting. And I really thank my co-chair, Sarah, and all the panelists for their great contributions. And I wish them extraordinary success in developing these new therapies and bringing them to all the patients, and all the best of luck for the following period. Thank you very much.
Thank you, Esteban.
Thank you.
Thank you.
Thank you.
Thank you.